Welcome to my talk. I've titled it Riccia, Small Plants, Big Genus, and it's going to be a little bit of an unconventional talk. No real methods or results, but more of like a educational overview of liverworts in Australia as a genetic resource. So yeah, if you're familiar with us down in Melbourne, the Monash node, in John Bowman's lab, we do developmental genetics on Marchantia polymorpha, which is a liverwort. And we'll go into more about why we do that. But yeah, today I won't be focusing so much on Marchantia polymorpha and more so other species that we have access to, majority being in the Riccia genus. And partly an explanation of what this talk is about is that in Melbourne, after 2 years of lockdown, we just finished the Centre of Excellence Hobart retreat and the COVID isolation that we did following that. We had a rainy winter, which is good for growing Riccia. And we really just wanted to get outside and pretend to be botanists for a little bit. So if you've heard of the saying, touch grass, I think we morphed that into go outside, touch a liverwort, mental health sort of thing. And so the rest of the talk is sort of a post hoc academic rationalization for collecting so many Riccia. So there's some Riccia there. So in the Bowman lab, we're known for promoting Marchantia polymorpha as a genetic model, and I think it's been quite successful, and it was a good choice. But some of the reasons why we use Marchantia polymorpha is that a lot of things are very simple in Marchantia. One, it's got a small genome, it's got very few genes, and for all the gene families, there's only single copies of certain genes. If you knock them out, you can very easily see effects in the plants. You can easily clone these plants. You're also very easy to transform genetically. They sit in a key evolution position where we can make comparisons to Arabidopsis and therefore all of land plants, and also make some inferences about what the last common ancestor of all land plants look like. So yeah, quite powerful for evolutionary looking at very core gene functions within plants. They're also globally distributed, which is quite important because, a lot of us are plant scientists in Australia, so we know that biosecurity does suck a little bit for Australian research. But yeah, you can find Marchantia growing as a weed in Bunnings, so that's quite useful. So I'm just going to start with the evolution of liverworts and the phylogeny, because that's going to be important to understanding where Riccia sit in relation to Marchantia. And so here's the very simple vision, or the simplest vision we sort of give of Marchantia. So you've got way back 500 million years ago, you've got freshwater green algae. At some point, they've come onto land, and very quickly you get this diversification of land plants, and you get this two major branches, one being the one of the higher plants with Arabidopsis on there. So they have multiple innovations, increasing complexity, creating all the interesting plants, like your crops and everything. And then you've got this evolutionary backwater of primitive plants, which has Marchantia polymorpha on it. And this view is, very simple, so it's easy to tell someone very quickly. But obviously there's some misconceptions that come from this. And one of the misconceptions, that I'm going to address today is that sometimes we think that Marchantia polymorpha looks like the last common ancestor of land plants, in that it hasn't changed since it split off from the rest of the plants. But of course, as with everything that's surviving today, it's gone through the same amount of time of evolution. So if we make this tree a little bit more complex, so yeah, still down here, you've got lycophytes, fern, and seed plants, which is, the major groups in the vascular plants. And then over here, you've got the bryophytes, so that's the mosses, which people probably recognize, liverworts, which has Marchantia polymorpha in it there, and the hornworts, which is even less known than liverworts. So all of these plants, they have evolved in the same kind of environment that the first land plant might have lived in, but they've all diversified to some extent. And so we're going to be looking here at Marchantiopsida, which is again, what contains Marchantia polymorpha. And so we'll just increase complexity again. So in Marchantiopsida, you've got a few different branches here, but then you've got the Marchantia genus and you've got Marchantia polymorpha over here. And so you've got a number of innovations going on here. So very early on in the liverworts, you've got this evolution of dioiky. So it's just having a male plant and a female plant, and you've got this dimorphic sex chromosomes, which are U and V sex chromosomes evolved here. So very old sex chromosomes. And you've got these cells that have oil in them called oil bodies. You've also got gemmicups evolved, so they help to clone the plant and spread it around. And you've also got air chambers, which are a photosynthetic organ, and stalked gametangiophores for like sexual reproduction. And yeah, so there's a number of innovations that have sort of happened. And so the dinosaurs might have seen something like a modern liverwort with like the little umbrellas coming up and everything. But back when plants first came onto land, certainly there would have been none of these features I was just mentioning, like the stalked gametangiophores, etc. Although this is in the gametophyte generation, and the sporophyte generation has actually simplified within the Marchantia lineage, so the sporophyte is less complex in Marchantia than it is back further back in time. As this talk is about uses of liverworts apart from Marchantia, I'll also mention that here we've got Marchantia polymorpha ruderalis, which is a subspecies that we have the genome sequence for and we do our work on for developmental genetics. But there's also Marchantia paleacea and Bertruana over here, Bertruana being an Australian species that can be useful because just here, polymorpha lost mycorrhizal interactions. So if you want to study ancient mycorrhizal interactions, you've got to move out to these very closely related, but not quite the same as polymorpha plants. And another plant that's in Australia is Marchantia foliacea. And we're thinking of sequencing that in order to get a good, you can find conserved non-coding elements if you get a plant that's roughly this distance. So it's the genome of this could be useful for us doing things there. But the main part of today is on this little branch here, which I haven't expanded out yet. So you've got all these other genus here in liverworts, and here is Riccia. Oops, sorry, I've got to go back. Accidentally clicked. So yeah, here you've got Riccia, and it is the largest genus within Marchantiopsida, having about 200 species. But yeah, this whole group here that Riccia is in, I was saying that you've got this evolved features, new innovations coming up here, but actually in this group here, you actually lose your gemmicups. Some of them have lost their air chambers, and in the Riccia, they've lost their stalked gametangiophore. So they've sort of re-simplified. Riccia are just a very diverse species thing. And one of the important things here is that, there's 200 species, but like 50 or so of those species occur in Australia. So we've got access to a lot of these Riccia plants. So again, just more introduction. Here's the life cycle of Marchantia polymorpha. So we'll just quickly go through. You get spores. Spores will grow up into this flat green thallus, which is the main body of the plant. Marchantia polymorpha can be cloned through from gemmicups, or it can go through sexual reproduction with separate male and female plants. You can have sperm swimming through water up into this little umbrella where there's eggs. Eggs get fertilized, and this sporophyte develops underneath this umbrella, nutritionally dependent. And when they're mature, they'll pop out on this setter here, and the spores will be wind dispersed. So Riccia is simplified, as I mentioned before, and so you don't have these gemmicups, you don't have these stalked gametangiophores, and the sporophyte stays within the thallus, and so it doesn't have this stalk on the sporophyte itself. So here's an actual picture of Riccia. This is Riccia crystallina, and actually, you can find this just in the nature strip in winter out in the suburbs. So you get the spore grows into this small, only a few millimeters across green tissue. And that happens in winter when it's sort of wet and not too dry. And then in spring, this Riccia Crystallina specifically will dry completely out and die, but inside the tissue, you've got the spores in the sporophyte here that are growing, and yeah, these spores will stay there and presumably germinate again the next winter. And here's a size comparison with Marchantia polymorpha spores. So the rest of, well, not the rest, there's a whole series of slides coming up where this is just a slideshow showing the plants. And so just so you can have a look at what they look like. So here's one species of Riccia, and I'm just going to go through a bunch that I've managed to photograph. And yeah, you want a close look at these. I do also have them up on my Instagram, but just so you get an idea of the diversity of form you can see with the Riccia. And okay, nearly done. Okay, so we've collected a whole bunch of Riccia. We have them in the lab and we've been growing them. And now we're thinking, okay, well, so what actual science can we do with these plants? Obviously, we did have some ideas what we could do before we collected them, but we are still looking at things we can do. So the first rationalization for collecting Riccia is that Marchantia polymorpha has the oldest sex chromosomes that have been, I guess, studied so far, diverging from autosomes about 430 million years ago. And so for comparison, our X and Y chromosomes are only 40 million years old. So that's like, an order of magnitude difference in how old these chromosomes are. But if you go away from Marchantia polymorpha, so here there's this outgroup of Riccia is this Riccia carpus natans, which she'll be seeing in our lab, help do the genome for as part of her thesis. So Riccia carpus natans doesn't have separate male and female plants. It's just got male and female in the same plant, and it has lost the U and V chromosomes. And so part of sequencing Riccia carpus natans was to figure out what has happened to the U and V chromosomes in this plant. But within Riccia itself, you've got both male and female plants separate and together. So this is monoicous and dioicous, but you can see with the symbols here. So potentially one option is that there's a monoicous common ancestor here, and that you've got multiple transitions to dioicy with these species here, Riccia collata and Riccia fluitans, and there's many others. Or it could be the other way around. But really, if we just need to sequence quite a few different Riccia with different conditions, and we can sort of ask some more questions about what is going on with the sex chromosomes in these plants. So depending on what situation we find, you can look at what genes are conserved when you lose your sex chromosomes and find out more about what genes are important for the sexual dimorphism. And we can also ask questions about the transitions between dioicy and monoicy and what happens in chromosomes, say, when you make new sex chromosomes or when you're multiple independent losses of the same sex chromosomes, whether it happens the same way each time. So another thing we can look at is aquatic liverworts. So again, with Riccia carpus natans, because I guess we started working on that first, and Riccia is sort of newer, but in Riccia carpus natans, it is a aquatic liverwort in that it floats on top of a pond. And so these scales here are under the water, and this is the surface. Oh, keep on clicking as I'm zooming. And this surface is above the water. So a little bit like duckweed. But as the water dries up and they go onto land, the scales stop being produced and it produces rhizoids. So there's a developmental transition there. And if you supply the plants at ABA, you can get the same transition to happen without the loss of water. So there is a genetic network conservation there, which is interesting to look into. But if we add Riccia into the equation, there's been multiple independent evolution of aquatic liverworts happening. So most of the Riccia aren't aquatic, but there's at least 3, maybe 4 or so that have evolved to become an aquatic. And so, yeah, again, we could look at these plants and see if there's similar gene losses are happening if we just do a sequence of the genes they have, or if we do, we could potentially look at it multiple copy numbers if we do more deeper sequencing. And And here with Riccia fluitans, a group in Germany has even managed to transform it. And so they can do some more developmental questions because they can do some more genetics on that plant. And we could do the same here with Riccia carpus natans. So another thing about Riccia is that a lot of the species that we've collected are actually new species. And so partly that's because we've been getting help from someone at the Australian National Herbarium, Christine Cargill. And so she's been saying that the species that she's sending us, she knows aren't classified species yet. And one of the reasons for this is that, say here with Lamellosa, but also down here with these two of labeled inflexa, is that some Riccia have been classified as the same species because they look the same, except then Christine's done some DNA marker analysis and found out that they're actually polyphyletic, meaning that these plants must have converged upon the same appearance. And so one benefit, again, we're not botanists, but if we did do some sequencing, we could help resolve the phylogenetic tree of Riccia. And all that these are, obviously Australian species, so that's a good thing to do to help Australian research and conservation of, I guess, these interesting little plants. But also this strong convergence could also have a strong signal within the genome, and we could look at how different genes, potentially developmental genes, are converging in these plants. So again, this is Riccia inflexa, which I was showing before on the other slide. And so if you think back to Vanessa's talk, if you, I don't know if we necessarily have cavitation because we don't have vasculature, but if you dehydrate out most liverworts, you dry them out, they will die. They won't bounce back from drying out, but there are quite a few that show resurrection. Resurrection being they dry out, you water them again, and they come back nice and green. In this instance, you can actually get plants drying out for like 10 or more years and then coming back from being completely dry to resurrected. In this case, this is 15 minutes after being watered. And there's multiple, as I was saying, multiple species in liverworts that have evolved to do this. And one common feature you can see is that this black edging around the sides of these liverworts. And so the idea is that as the liverwort dries up, the bottom surface curls up and over, and the black scales on the bottom protect it from harsh sunlight and things like that. And so, yeah, potentially there would be signatures of this within the genomes of these plants. But again, this might be a little bit harder to analyze in a genome. Someone at a conference recently suggested that they would be looking at the endophytes, so like fungi and bacteria that are living on these different liverworts, and if they've got the same sort of bacterial makeup, because they could be inferring some of this resurrection or drought resistance tolerance, which again that is interesting. Obviously not something we have expertise in, but it is cool. And so here's another kind of silly thing that I've found because we've been growing a lot of these plants. So when you scratch the surface of this Riccia inflexa here, you get a really strong smell like cut grass. And at least I think when you do the same thing for this plant over here, Asterella tenera, it smells a bit like fish. And so again, you could have these chemicals could be converging on a similar structure to something found in other animals or plants, or it could be some sort of conserved chemical processing. But either way, I think a lot of liverworts probably do have novel secondary metabolites that aren't well researched, and maybe if we do sequence the genomes of these plants, there could be some use for other people to look into it that way. But also I'll just point out that I've given this slide a very fancy title with lots of scientific words just to compensate for the fact that this is just a very fluffy, fun thing that I just scratched the liverworts and they smell interesting. So a little bit more sciencey, I've done a preliminary analysis of spore dormancy within Riccia. So for context, Marchantia polymorpha spores, there are thousands of them, and we use those for transformation. So we grow the spores up into sporelings, and then we add agrobacterium and to get genes into them. But they basically have no dormancy. So all the spores we put out, the thousands of them, they'll all germinate, and then we could have thousands of attempts to get the genetics into them, and it's all very efficient. Well, even if it's not efficient, it doesn't matter because we've got so many spores. But with the Riccia, basically they all have dormancy, and I did a few different environmental treatments, so like heat treatment, cold treatment, soaking in water or abrading them in a vortex to see if I could help to break the dormancy and get them to germinate more readily. And this worked to some extent in some of them, but most of them still retain their dormancy. And so you can see here for this one, it had a peak with getting heated, and this one here, the most of them germinated under cold soaking overnight, but still very low numbers. And so it'd be quite interesting to look into dormancy as a developmental phenotype potentially, like maybe how is dormancy being regulated? Is it similar to like in higher plants or seed plants where say you've got things like vernalization and methylation going on there? How is dormancy controlled in these spores would be interesting. Of course, how we do this would be a little bit complicated. Potentially might have to do some RNA-seq or something on spores or check the methylation, but still could be good. So I suppose I'll end it there. I do have some, I guess, some concluding thoughts. Just the main point here is that Riccia are very diverse. We've collected a bunch of them. Potentially we'll sequence their DNA or RNA, and we can look at things like convergence with sex chromosomes, the aquatic habitat they live in, their resurrection ability. We could help identify new species, and just generally look at plant development from this different angle. And so, yeah, so basically, we've got Marchantia polymorpha, and we've been using that to get new perspectives on flowering plant genetics. But now we've got Marchantia polymorpha, genome and all this knowledge there. We can use that as a stepping stone to explore other liverwort diversity. Yeah, so anyway, I hope it's been interesting.